Thank you, Professor Tamura, for kind introduction. My talk today is describing the destiny of antibiotic resistance genes after releasing from the veterinary and human environment. Dr. Tamura mentioned at the first that we are standing against the antibiotic resistance according to the WHO concept, One Health. The One Health includes the health of people, health of animal, and also the environmental health. So I'm starting my talk from the reality of microbes in the sea. Probably most of you might not be familiar to marine bacteria. So I will show some basic knowledge in the first section. And followed by the example of specific distribution of ARGs in seawater, especially Sargent experiment. And the second experimental result is formation of background of ARGs in environment. And I will conclude the linkage of human activity and aquatic systems. This shows the dynamics of antibiotic resistance genes, ARG. We have a hotspot, for example, in hospital, where contaminants of the antibiotics and the antibiotic resistance genes. And then we have another hotspot here, the pharmacy manufacturer. Especially recently, we have generic medicine wastewater, which is a very serious problem in the world. And then, of course, we have another hotspot in our community. So from these hotspots, we have wastewater, and the wastewater is going to the wastewater treatment plant. But this plant cannot decompose the ARG and drugs completely. So we have another hotspot in here. As you know, the animal and vegetable farms are using the much amount of antibiotics and antimicrobials compared to human medicine. And then sometimes we have wastewater directly to the water environment. And also we have another hotspot, aquaculture, where the antibiotics are used. So in any case, we have effluent from various settings, which goes to the sea. And here I have a question. Are ARGs in the sea increasing, decreasing, or constant? And also we have to know the process of gene recombination. And risk to human are also our study aim. So first of all, I would like to show the reality of microbes in the sea. Ecologically and physiologically, marine bacteria is quite different from animal bacteria. So here I showed a number of prokaryotes in our planet. Most of the microbes are living in ocean subsurface and terrestrial subsurface. So they cannot meet us forever. And then here you have soil microbes here, only 4%. And then here, aquatic habitats has only 2% of microbes in our planet. The aquatic environment contains all seas, all rivers, all groundwaters, all lakes, and all glaciers in the world. But we can meet them sometimes. So aquatic bacteria is closer to our life compared to other environmental bacteria. So this is the diversity of marine bacteria and archaea. If you survey the database, the half of the microbes, half of bacteria, half of the prokaryotes are proteobacteria. Among the proteobacteria, the alpha proteobacteria and the gamma are the measure in marine environment. In open sea, alpha is measured, and the coastal area, we have gamma as a major group. And then marine environment does not have, the gram-positive bacteria are very rare in marine environment. In physiological characters, the marine bacteria are cyclophile, oligotroph, and halophilic. And sometimes we have a piezophile and a thermophile bacteria, which are living in the deep sea ocean. And this is a species number. The seawater has this number, over 10 to the third OTU, which is almost equivalent to the sewage, soil, and human intestine and saliva. So seawater contains a large number of species. But the specific character of marine microbes are most of them, more than 99%, are non-culturable or yet to be cultured. So if you want to isolate drug-resistant bacteria, you can only see the 1% or less, okay, of the bacteria. So next section, I will show the example of a specific distribution of ARGs in seawater. Which ARG are targeted? If you have the veterinary drugs, the tetracyclines are the major, and then penicillins, and then here yellow color is sulfonamide. So the worldwide trend, tetracycline, penicillin, and sulfonamides are the major drugs. In case of Asian developing countries, sulfonamide is much more higher, larger percentage. So TET genes and SAR genes are appropriate target to chase the gene dynamics in environment. So this is my research. Specific distribution of SAR genes in the Philippine waters. Here you have Metro Manila in the center of this map. And then this Metro Manila has a very huge freshwater lake, Laguna Lake here, which has many aquaculture pen net. And also this lake, both coasts of this lake has chicken farms and pig farms. So wastewater from such farms go to this lake. And then water of this lake is going to this river, big river, and then go to the Manila Bay. So we examine the surface water from lake, river, and bay, seawater. So this is a strategy to survey antibiotic resistance genes. As I mentioned, more than 99% of marine bacteria are yet to be cultured. So, so my hypothesis is that the yet to be cultured bacteria are also ARG reservoir, and they can transfer the ARGs among the communities. So we took sample waters, and then this left side is a conventional culture method. We count the total cell number with DAPI staining, and then we did culture on the agar plate. And also we cultured bacteria with and without target antibiotics. And then we scrape all colonies from the plate, and then we purify the DNA from all colonies, which is culturable bacterial DNA. And also, we filter the sample waters through 0.2 micrometer nuclear pore. From the filter, we purify the all DNA, which is natural assemblage DNA. And then we did qPCR to know the copy number of SAR genes, and then normalized with 16S ribosomal RNA gene copy number. And this is the result. When you look at the natural assemblage, the vertical axis is copy number, and then horizontal bar line is sampling site. Here, from lake to river, mostly you cannot get any SAR genes. Here, SAR 1, 2, 3, almost nothing. Although the 2 sites we can get positive. But when you look at the C sites, we have positive for SAR 1, 2, and 3. So all SARs were detected in natural assemblage in the sea. And when you look at the culturable bacteria, the profile, the character is quite different. Here, from lake to sea, you have a positive result for SAR 1 and 2, but not for SAR 3. So this suggests that the culturable bacteria possessed only SAR 1 and 2, not SAR 3. So this suggests that yet to be cultured community in the sea is a reservoir of SAR 1, 2, and 3. SAR 1 is SAR 3 is known that not abundant in clinical settings. So that we are thinking that SAR 3 might be marine origins. Recently, the quite new SAR gene, SAR 4, was found in deep sea ocean. So I think some unknown genes are still in marine environment. So here, who possessed the SAR genes? You cannot see any species number. That doesn't matter. You just recognize the color. So the red color species are the natural assemblage bacteria. So from cyanobacteria, actinobacteria, and alpha, beta, and gamma proteobacteria are included in natural assemblage. This indicates that the seawater contains a variety of phyla. But if you culture, you can get only green colored species. All of the green color are gamma proteobacteria. So the gamma proteobacteria possess SAR 1 and 2. And then diverse members in the sea possess the SAR 1, 2, and 3. So it reminds us that possibly various marine bacteria possess the various unknown ARG. So here, I will talk about formation of background of ARG in environment. If you learn the marine food web in high school biology, you will learn this line. Starting from phytoplankton, and then go to zooplankton, and finally fish. All biota, all living things in this linkage are particulate organic matter, particulate living things. You can see all living things by your naked eyes. Okay? But we have another food linkage describing here, which is called microbial loop. This loop is starting from dissolved organic matter, DOM, which is coming from all biota, all living things. Dead body and excreta are decomposed and come to the very tiny organic matter, DOM. And then DOM can be used by heterotrophic bacteria. Only bacteria can use the DOM. So the bacteria is very small, but it's already POM, particulate. So then, once you get the particulate, the protist degrades the bacteria. Okay. So if you think about the biomass of POM, POM includes all living things, including bacteria. Okay. It's POM is 3 petagram carbon in the world. But if you look at the DOM, it is 700 petagram carbon. So it is very clear that living things are very minor organic matter in marine environment. Okay. So the bacteria is a very huge motive force to transport the DOM to higher eutrophic level. So when the terrestrial bacteria comes to the ocean, they should be grazed as well as the marine bacteria. Okay. So what happens when bacteria are grazed by protists? in microbial loop. The grazing. Grazing, the ciliate and the heterotrophic nanoflagellates are very important consumer of bacteria in aquatic environment. So of course, terrestrial bacteria are also grazed, as I mentioned. In this step, the DOM should be decomposed. But it is known that not completed, the grazing cannot decompose the DNA completely. So DNA is released as cell-free DNA, extracellular DNA, which supplies ARG into the environment. So here, my question is, are these extracellular ARG in dissolved fraction in environment are stable? So to give answer, we did microcosm experiment. We used the bacteria having the multi-drug resistance gene, resistance plasmid, the Photobacterium damsera strain YA. And then the plasmid name is PAC1. And then grazers are 2 species of ciliate and 2 species of HNF. We prepared monocultures, which include bacteria only, ciliate only, and HNF only. These are the control groups. And then we prepared the co-culture group, which contains the bacteria and the ciliate, or bacteria and HNF. And also we prepared drug containing group to see the effect of tetracycline. And then we sampled with time course, and we did a quantitation of cell number and ARG copy number. We monitor the TET-M gene copy because this bacteria has only 1 copy of TET-M. So in this result, if you have YA strain only, bacteria only, the bacteria number is not changed through 20 days. And then bar graph shows the TET-M copy number. Here, no change. But from initial, you have a positive TET-M copy number here. So we are calling this amount as a background. And then if you put the ciliate, the bacterial number is decreasing very quickly. And then at the same time, the TET-M copy number is increasing. And then the copy number is decreasing gradually, but it takes 1 week to get the background level. So always we have background level here. And then when you use the HNF is very small protist compared to ciliate. So the grazing rate is very slow. So it takes time to get decreasing of bacteria and then increasing the TET-M releasing. So the released TET-M decreased very slowly. It takes a week. And then OTC, in this case, OTC did not affect any in any groups. So this indicates that the marine protists accelerate the release of TET-M to environment. And then certain amount of TET-M, ARG, is always present as a background level. So this is a result of the residue of plasmid and the absence of selective pressure. We put the bacteria into PBS and then look at the increasing or decreasing through 30 days. And then when you add the tetracycline here, the total bacterial number is not changed in any case. We are using the 3 systems. One is a PAC1 in Photobacterium, and then this is a E. coli transconjugant with PAC1. And then right hand is ink FIB plasmid in wild type E. coli, which is isolated by Usui-san from the cow feces. In this case, the colony forming number is decreasing like this. This shows that the bacteria is going to the viable but non-culturable state. So colony formers are very decreasing. In this case, in any case, the plasmid number is stable. So this shows that if you do not have selection pressure, the plasmid is very stably retained in the bacterial community. So this is caused by PAR-AB genes, which are needed for the proper segregation of low copy number plasmid during cell division. If you have a PAR-AB, the plasmid can be separated equally to daughter cells, even in the selection pressure is absent. Okay. So we said ARG are easy to get and hard to lose. So here I summarize the two pressures. If you have a selection pressure case here, and then usually if you do not have the selection pressure, some plasmid is lost. But in our case, like in our case, if you have a plasmid with PAR-AB gene, the plasmid is retained. Okay. Plasmid persists in the community, even without antibiotic, selection pressure. In case of grazing pressure, until now, this is common sense. If the protist grazes the bacteria, all organic matters are decomposed. But our experiment shows that some antibiotic resistance genes, plasmids, survive after grazing, after digestion. So the plasmids are relatively stable in environment with and without pressures. So I will summarize the linkage between human activity and aquatic systems. You have a hot spot here and here, and then wastewater is going to the wastewater treatment plant, and some are decomposed and diluted in water, but some are stable in ocean. So the silent majority, the non-cultured, yet to be cultured bacteria, are the huge reservoir in the sea. And risk assessment is needed for the ocean reservoirs. And this is the risk scenario that we excrete the wastewater to environment, and also veterinary STP, aquaculture, and pharmaceutical factory excrete the wastewater. And then in water environment, gene mixture and transfer are occurred between the culturable pathogens and also the yet to be cultured commensal bacteria. So if you have a chance to get the seawater, like here, we might have possible gene risk and also exposure risk. So when you think about the One Health, we should think the circulation of ARGs among the human, animal, and the environment. So it is needed to see environment to make clear the ARGs circulation dynamics. Okay, thank you very much. This is my team members, and also I would special thanks to Miyari-san Pharmaceuticals for invitation to this symposium. And then these many grants are supporting my research. If you have a time, please visit my city, Matsuyama, very beautiful city. Thank you very much.
Thank you very much.